successfully record. Sometimes they don't successfully record. It just depends on the day. So just give me a few seconds. I am trying to share my screen. Okay, there it is. You should be able to see a whiteboard in less than 5 seconds. You should be able to see a whiteboard on your side. There is a mic that is on. Is someone asking a question? Okay, so I'm going to try and mute everyone. Okay, so please thumbs up if you can see my whiteboard, just to make sure we're on the same page. I'm seeing quite a few. All right. So if it vanishes any second, please unmute and say, ma'am, your whiteboard is no longer there. It's black or you can't see something, because from now on, I won't be able to see everyone. I will only be seeing my whiteboard. Now, I'm going to start by doing a little bit of background information. So the name of our topic is DNA. It's also called nucleic acid. So what we're going to try and find out here is just the basics of what DNA is, where it is found, how it looks like, who discovered DNA, what's the significance of DNA, and all of that before we get to the juice, before we get to DNA replication, before we get to protein synthesis, because we need the foundation of what this is before we get to that. Now, in order for us to understand exactly what this DNA is, I can, in simple terms, just say it's what makes you you as a person. This is the genetic information that makes you this tall person who is light-skinned or dark-skinned, who has curly hair, who is blood type O, who has blue eyes, who has whatever. So all your physical characteristics are determined by this DNA that you have in your cells. And now this DNA is genetic information that you inherited from your parents. So the DNA that you have in your body, you inherited 50% of it from your mother and 50% of it from your father. And the basics or the foundation of this topic is actually going to be important in meiosis and also in genetics. Now, when we say you inherited 50%, that's the word that you need to use. Don't use the word receive or whatever. You inherit this DNA because it's passed down to you by your parents. Now, the question would be, okay, where are we finding this DNA? So you're saying we have this DNA that determines my characteristic, determines your eye color, your skin tone, your hair length, your hair color. Every little thing that has to do with physical characteristics in your body is determined by the genetic information that we find in the cells. Where exactly are we finding this DNA? We find it in cells. So we as human beings have what we call cells as our building blocks. That's where it all started. So if we look at DNA, I'll come back to it. You will have cells which are building blocks. Like I said, sometimes the pen can be very slow. The cells, which are the building blocks of life. So there is plant cells and animal cells that are taught in grade 9. Now, we then say when you group these cells, you end up getting a tissue. And it's not the tissue that we use to blow our nose or to wipe when we're in the bathroom. But this is basically a tissue that is made up of group of cells. And these tissues, you can group them together to find an organ. So when we group the tissues together, we then end up finding an organ. And an organ can be your skin, can be your lungs, can be your eyes, and whatever. Now, in order for each and every organ to function, it will be because of the cells that make up that particular organ. So if your skin cells are able to detect temperature changes, detect feelings, you are able to feel all of that, temperature changes, pain, pressure, because of the cells. They perform their function for the organ. Then there's different organs that are going to work together to then form what we call a system. So examples of system, we can speak about the digestive system. This is a system. That's why I said the pen can be slow. So you will need to listen to what I'm saying instead of what I've written there. That is a system. For example, digestive system will include structures such as your mouth, working with your esophagus, working with your stomach and small intestines. Those are different organs in your body that enable you to break down the food that you eat so that it can be absorbed. Same with circulatory system, that's involving transportation of blood in your body with all other systems. I think in total, we have about 11 systems whereby organs work together and bring about certain functions. Now, when all of these systems work together, they form me and me and you are an organism. But where did it all start? It started with cells. So now let's understand what this cell is. And I'm going to focus on an animal cell, meaning these are cells that we find in animals, and me and you fall under that group. Now, these cells may be different for plants, and yes, they will still have other organelles that are the same, and other organelles that are different. So the basic structure of an animal cell is that it will have this membrane over here. Now, this is grade 8, I'm lying, this is grade 9 and grade 10 work. You won't be assessed about this in matric. However, you need the background information of cells in order for you to be able to answer questions on this topic. So the terminology that I'm going to mention here, you might think, this is not important. It clearly is, because we are going to be using these words when we answer in DNA and also when we do meiosis. Now, this line that I've drawn here represents a membrane, and we call this the cell membrane. It's the membrane of the cell. So I'm labeling it nicely. I'm not sure what I did here. I'm going to have to erase the second line. I'm going to try and follow rules, drawing rules, but anyways, if the pen is slow, I won't be able to follow all the rules. Okay, it's fine now. So this will be our cell membrane. So the cell membrane is a selectively permeable structure. This is the cell membrane. So please, if you can make a note of this, please also draw all of this on your side. Cell membrane. Okay, so this cell membrane is a selectively permeable structure. So it selects which substances go in and out of the cell. Because remember, for me and you to function, the cells actually in our bodies will need to function. And we are made up of millions of these cells. Then filling up the cell membrane is a jelly-like substance which we call the cytoplasm. Check where my line started. So filling up the cell membrane is a jelly-like substance where metabolic reactions such as cellular respiration will take place, and that is the cytoplasm. Now, this is where we will find different organelles here. So we'll find different tiny organs of the cell that will basically have different functions in order for the cell to survive. Just like in our bodies, we have the brain that controls everything. We have the eyes for seeing, we have the skin for feeling, the heart for pumping blood, blah, 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 blah. Those are our organs that allows our body to function every day. Now, within the cytoplasm, there's organelles that will allow the cell to function, to perform its function. Now, there is a few organelles here. I will mention the important ones for grade 12 only. I'm going to draw this one, which is the mitochondria. I'm mentioning it because it will come up in DNA and it does come up in evolution. So that is the mitochondria. 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 Thank you. There it is. The mitochondria is a tiny organ in a cell that basically makes energy for the cell. The main reason that I have energy to conduct this lesson is because of the mitochondria in my cells. The main reason you have energy to join this lesson and take down the notes is because of the mitochondria in the cell. And we have many of these structures per cell. As much as I've drawn one, you actually have more than 5 or 6 per cell. This is where cellular respiration takes place for the cell to use the glucose that we eat and the oxygen that we inhale in order to produce energy for us. Now, there's other structures in the cell. I'm just mentioning the ones that are important in grade 12. These little dots indicate what we call ribosomes, and we will speak about them in our topic. And I know you probably know about them already. If at school you have covered protein synthesis. So ribosomes. Then we have the brain of the cell, which I want to speak about. Remember, our topic is about DNA, and we're trying to find out where exactly do we find this DNA, right? So the biggest organelle that we find in a cell is called the nucleus, and that is known to be the brain of the cell. Just like in your body, you have the brain that controls the brain is controlling everything about my movements, what I'm saying, and all of that, what I'm able to feel. It's all controlled by the brain. Now, the brain of the cell is the nucleus. Now, we are going to focus on this because it is very important. We normally find it in the middle of the cell. And this nucleus has its own membrane. And what I'm drawing here is the membrane of this nucleus. Very important. So I'm drawing this membrane more like dashes. It's not a circle. And the reason for that is because there are spaces or pores in between. So if I'm labeling it, I know I'm breaking rules. If you know biological diagram rules, I'm probably breaking them by doing this, but I'm going to label this as your nuclear membrane. Nuclear, yes, membrane. Membrane. Okay, that is membrane there on top. Okay, these are the lines that I've drawn here. Now there's two words that have the word membrane. Remember when I started drawing the cell, I said you have the cell membrane. That is the membrane of the cell. And then over here, I said you have the nuclear membrane. That is the membrane of the nucleus, which is what I'm drawing there. The brain of the cell. It controls all activities of the cell. Now, these spaces between the nuclear membrane are called nuclear pore. They are quite important for us to know because when we do transcription, protein synthesis, there is going to be a molecule that leaves the nucleus through the nuclear pore, and we need to know that. So just going to decrease the thickness of the pen. Yes. Okay, so this will be the nuclear pore. Okay, you have the correct spellings in the notes. If you can't really see this, nuclear pore is Nucleo. Nucleo. Either you say nucleo or nuclear pore, it doesn't matter, they'll still give you a mark. Okay, so these are the spaces in between the nuclear membrane. Then listen to this. Filling up the nuclear membrane is a plasm. Now, remember we had a plasm that filled up the cell, which was the cytoplasm. The plasm that fills up the nucleus is the nucleoplasm. The word sounds very, very familiar, right? Plasm, plasm, plasm. They sound the same, but they are not the same. Cytoplasm is the plasm of the cell, C for cell. Nucleoplasm is the plasm of the nucleus. So this will be the nucleoplasm. Okay. Oh my God. Nucleo. That is a Plasm. Okay, that looks better. That is the nucleoplasm. That is the plasm of the nucleus. Now, in the plasm of the nucleus, we get the DNA that we are talking about. And normally in this form, it will be in an entangled network, which we call the chromatin network. So that is an entangled network of the DNA that determines all the characteristics about you. And we call that chromatin network. It is inside the nucleus. So if someone were to ask you, where do we find this DNA? It is inside the nucleus of a cell. You don't find it floating anywhere else. So this will represent the chromatin network. Chromatin. Come on. Tin network. There it is. All right. Now we're not done with labeling or putting information about the nucleus. There is something else in the nucleus, and we normally represent it with a big dot in the nucleoplasm, and that is the nucleolus. Now, the nucleolus is where we find a different kind of a nucleic acid, which is RNA. And then we also find proteins of the cell. So proteins are basically organic compounds that are needed by our bodies in order for them to function. So we need them to produce our cells, we need them to build our muscles, we need them to form hormones and enzymes in our body and all of that. And this is the last structure that we are going to label nucleolus. I'm going to project the slides after so that at least we get the correct spellings for everything, since you can't really see the words. For example, chromatin network, it's not nicely written there. So this will be the nucleolus. I'm sure you can notice something about all the labels for the nucleus. They start with N and they sound the same. Nuclear membrane, nuclear pore, nuclear plasm, nucleolus. Two words to not confuse. Cell membrane, membrane of the cell. Nuclear membrane, membrane of the nucleus. Cytoplasm, plasm of the cell. Nucleoplasm, plasm of the nucleus. Now there is our DNA. We have identified the location of our DNA. Now give me a second. I'm happy to take questions because I'm about to project the notes now. So if you have a question, please do ask me before I go to the notes. Okay, no questions. So please give me a second. Okay. In 5 seconds, you should have slides on your screen. So just give me a second. I'm sorting them out. 1, 2, 3. Okay. Okay. Yes. They should be on your screen now. Thumbs up if you can see the notes. Let me see. Okay. So I guess you guys are still waiting because I'm not seeing any thumbs up. Okay, thank you so much. All right, let the work begin. All right, all matric notes have this revision about the nucleus. I was just giving you the background. So all of the terminology that I've mentioned, there it is. This is the nucleus. Now we are no longer seeing it in a cell. Remember in my diagram, we had a cell first and then the nucleus. Now we are just focusing on the nucleus. Okay, so what do we have here? We have the nuclear membrane, which is also called nuclear envelope. So in a test, whether you say envelope or membrane, you will still get a mark. And there are spaces in between this membrane. We're able to see those spaces, and I said they are called nucleopore or nuclear pore. Then the entanglement that you find inside the nucleoplasm, that is the chromatin network. You have it over here, chromatin network. If you just write chromatin, you won't get a mark. I think network was just removed here by mistake. Then the plasm filling up the nuclear membrane, that is the nucleoplasm. Then there is that big dot, which is the nucleolus. The endoplasmic reticulum, yes, you have it in our diagram, but it's not important for grade 12. Those are just extensions of the nuclear membrane, and they have ribosomes attached to them. So all of these red dots are basically ribosomes where proteins are made. Now, let's look at this, me and you, and we are going to do it quickly. So in an exam, they're not going to ask you about the nucleus in this form, but nucleus terminology is very important. So A, that is our nuclear membrane, we're able to see it. B, that is the pore. C, that is the nucleolus, sorry. D, that is the ribosomes, these dots over here. Then E, that is the nucleoplasm. F, that is endoplasmic reticulum. G, that is your chromatin network. And H, that is the cytoplasm. So it's not pointing anywhere. There it is over here. So that's the plasm of the cell, because that's where we find the nucleus floating. And I, it's the nuclear membrane. So easy peasy stuff. So this is all the information I've mentioned about the nucleus. This slide is super, super, super important. Now, the functions of the nucleus, there's only 3 functions, and they normally do ask. And the unfortunate part here is that they don't allow you to substitute the words. If they use the word regulate, you actually need to say regulate the metabolism. You can't say something else. It tries to balance the metabolism. You're not going to get a mark for it. So marking life sciences is very unfair. They are quite strict in how they want the learners to answer. That's why it's so difficult to get 100%. You do have learners getting 94, 96, 98, but it's not easy to get 100% because of the marking. So in terms of functions, one of the functions of the nucleus is to regulate the metabolism of the cell. And the tick, guys, will be here at the end. You can't just say regulate the metabolism. In life sciences, you write full sentences. If you don't write full sentences, you won't get a mark. So this nucleus, since it is the brain of the cell, it regulates reactions or cellular activities that take place in the cell. If you don't mention anything about the cell, you won't get a mark. If you don't want to say it regulates the metabolism of the cell, you can say it controls all cellular activities of the cell. 2, you can also say it regulates the structure and functioning of the cell. So it controls which substances are produced in the cell and the structures there in the cell. Also, it carries our DNA. The nucleus is where we find the DNA, which is our genetic material. So it carries the hereditary characteristics of the cell, or it contains the hereditary material of the cell. So these are the 3 functions of the nucleus that you need to know. Please take note of the terminology that is used here. Regulates metabolism, regulates structure and functioning of the cell, carries hereditary information or hereditary characteristics. That is what is important. Okay, we're going to skip this and then skip that. These are just diagrams. That's it. Then we come here. When we speak about DNA, we need to understand, yes, we know we find it in our cells, but actually what is it? It's considered a nucleic acid. So nucleic acid are organic molecules. There's 2 of them. There is a DNA and then there is an RNA. So if you want to know what these words stand for, you can study it, but there is no one that is going to ask you in matric to tell us what DNA stands for or what RNA stands for. You just write that in a test and you get a mark. Now, DNA stands for ribonucleic acid, deoxyribonucleic acid, and RNA stands for ribonucleic acid. Now, we are going to find out information about these nucleic acids. Both of them, you find them in the nucleus of the cell. If you remember in the drawing, I said this is the nucleus, and then you'll have the chromatin network, which will contain the DNA. Then there is a big dot inside the nucleus still that contains RNA. Now, as much as in our diagrams, these look very small, but they're actually extremely large molecules. And large molecules are called polymers. So I normally teach this way. So a large molecule, which is a polymer, there is the terminology here. That's a house. You think of a house. But remember, you need to build a house. So you need something to build a house. If we want to build a house, we are going to need building blocks and cement to build that house. And if we're to break down that house, we'll break down that house and still get the building blocks. So in this case, the polymers are these big houses, but big houses are made up of building blocks, and the building blocks are called monomers. So what you will then have is, a building block, which is a block there or a brick with another brick, and then you put them together with cement, obviously, in order for you to get a big house. The house is the polymer and the blocks are monomers. Now, if we're then to apply it to our nucleic acids, the nucleic acids themselves, DNA and RNA, are the big houses, meaning are the polymers. Now, we are going to need the building blocks of DNA and RNA, and those are called nucleotides. So we have nucleotides to build DNA and RNA. So the monomer is the nucleotide, the polymer is the DNA and RNA. Now, they are made up of carbon, hydrogen, oxygen, and phosphorus. These are just elements that make up the nucleotides. This part of the slide is honestly not important. Now, the DNA and RNA are very important in our bodies. It's not just about coding for our characteristics, but they are also there to help with the synthesis of proteins, making proteins for our bodies. Yes, the normal protein shake that we buy and drink. Yes, the normal protein that we get when we eat eggs and meat, that protein is very important for our body. So what are we getting from this slide? We are getting that we have two nucleic acids, DNA and RNA, but these nucleic acids don't just come from the air. We need to build them. They are polymers, and they are built by having what we call nucleotides. So a group of nucleotides will form an RNA and will form a DNA. So this is explaining it. So you'll have a monomer, just one thing here, then they get grouped together, they form a polymer. Then, where exactly do we find DNA and RNA? This is important to note. We find DNA, number one, in the nucleus. So in terms of DNA locations, we find DNA in the nucleus. And the DNA that we find in the nucleus is called nuclear DNA. That's the one that you find in the chromatin network inside the nucleus. And it makes up chromosomes. We will speak about this at a later stage. The second location for DNA is the mitochondria. Remember in the cell, I drew the cell and I said in the cytoplasm, you have the structure that is responsible for energy. And I said that is the mitochondria. Now you also find DNA in the mitochondria. Please note this. If you don't want to write the full name for mitochondrial DNA, you are allowed to write mtDNA. You will get a mark for it. However, you need to follow the right format. Your M and your T will need to be in small letters, and DNA will need to be in capital letters. So this will be correct. But if you were to write it like this, M capital letter, T capital letter, and then DNA also capital letter. My slide is moving. I'm not sure why. Okay. So mtDNA, this would be incorrect because your M and your T are in capital letters. So if you want to write mitochondrial DNA by just abbreviating it, your M and your T will need to be in small letters. Then we also find DNA in an organelle which we call a chloroplast. So this is an organelle that we find in plant cells only. It's not really covered in metric, so we are going to leave it at that. In terms of RNA, we find RNA in the nucleus. I've already mentioned this. In the nucleus, there is that big dot there that is in the middle, which we call the nucleolus. So that is where we find our RNA. We also find RNA in the cytoplasm, in the ribosomes. There is also RNA there. So now we know the location of DNA and RNA. Then we come to the history of DNA discovery. This slide is important. They will ask you for 2 marks somewhere in the multiple choice or somewhere in column A, column B, or somewhere in 1.4. They ask you about the history of DNA discovery. So all of this information that we are teaching today, it's not just information that we just woke up to. It was information that was researched long time ago by scientists. I remember when I was in high school, we needed to learn, I think, 2 pages worth of this information, and we needed to even be able to write an essay about it. And fun fact, the only thing that you need to know here is the names of the scientists that were involved. Nothing about the year. You don't have to worry about what happened in 1953, what happened in 1962. Don't worry about the year. It's all about the names of the scientists here. And there were 4 scientists. I'm going to summarize the story for you. There were 4 scientists that were involved in learning and researching about DNA, because the information that was out there in the 1900s was, oh, okay, I'm this tall because of DNA that I inherited from my father. I'm this light skinned because of DNA. But actually, how does this DNA look like? What is the shape of the DNA? What exactly are we finding in DNA? That's what they were researching. So on this slide, please make a note that you need to know the names of the scientists. They are important. The names of the scientists. Now, how we are going to analyze this, I'm not sure why my slides are moving up and down. How we're going to analyze this, we are going to say there were 4 scientists. And basically, there were 2 pairs of scientists that were working here. So we had pair 1 and pair 2 that were researching about the history of DNA or the structure and functioning of DNA. I'm taking out my pens here that are going to help me explain this. Okay, there you go. There were 2 pairs of scientists. I mean, we know how scientists work. They will work long hours to research something for years. So there were 4 scientists that were involved here. 2 of these scientists were working together in the same lab, and the other 2 were also working together researching the structure of DNA. So which pair is this? Rosalind Franklin and Maurice Wilkins form the first pair. In the notes, it doesn't say there was pair 1 or pair 2. That's incorrect. I am just teaching it like that so that you understand it. So the first pair was Rosalind Franklin and Maurice Wilkins. There they are. They are working in the same lab and researching about DNA structure. Then the second pair was made up of James Watson and Francis Crick. So these 2 will form the second pair. Now, what is important to note here is you need to remember the names and surnames of the scientists. However, we can give you a mark if you just write the initial and the surname. So for example, if you don't want to write Rosalind Franklin, you are allowed to write R. Franklin and then you get a mark. Now let's speak about the story between the first pair, Rosalind and Franklin. Now, as they were researching, this is how I'm imagining things, right? How things worked out. Now, there is no need for both scientists to be available in the lab for the same time. So another one can go to the field or wherever and research, and another one is not coming into the lab today or whatever. Now, Rosalind from pair 1 then found a DNA X-ray image. So he researched the structure, she actually was a female, she researched the structure of DNA and was able to get an image, which is called an X-ray image. So during the research, she was like, okay, cool, this is what I found. And remember, she was working with Maurice. And then Rosalind told her partner about it, Maurice, to say, hey, Fred, partner, I found this X-ray image that shows me DNA. But remember at this stage, it was not clear what DNA is made up of. So what she then did was to tell her partner. But let's listen to what the partner did. The partner, Maurice Wilkins, then took Rosalind's X-ray image and gave it to the second pair. Did not work with Rosalind to find out more information about the X-ray picture, took that X-ray picture and gave it to the second pair, which is James and Francis. It does tell us here that Maurice gave Rosalind's Franklin's picture to Jameson to James Watson without Rosalind's knowledge. So now there was a bit of a betrayal here. Rosalind found the image, told her partner, and then the partner took the image and gave it to another pair of researchers. So now James and Watson, who were also working on the structure of DNA, were like, oh my God, what is this? An X-ray image. And they were able to read it. They were able to say, oh, okay, what we're able to see here is a double strand. So we know we have DNA in our cells, but we don't know how it looks like. So they used that X-ray image and found out that DNA consisted of a double strand. Double means two. Now that means it's made up of two strands, and these two strands are paired with nitrogen bases there. They could only find out this because of the image that they found. Anything else in the slide, you don't need to know. But what is important to note is that in 1962, these three scientists, Watson, Crick, and Wilkins, were awarded these big prizes in science. They were awarded the Nobel Prize in Physiology for the work that they did in discovering the structure of DNA. But note how I mentioned three scientists and we started with four. Now, the reason for that is Rosalind died before she could be awarded any prize. And Nobel Prizes are not awarded after death. So she did not receive any credit for her work before she died. None whatsoever. Even the credit about the X-ray image, none whatsoever. So all the four scientists, we do teach about them in this day and age, and we speak about how they contributed to the discovery. But Rosalind died before she could be credited for her work. That's the story, and that's all you need to remember. The names of the scientists, though, are very important. Okay, please remember to unmute anytime and ask me a question. If you feel like I'm not explaining something or you need emphasis on something, I am here to explain. All right, we are coming back to these nucleotides. Remember we said when we build a house, we need bricks. And in our case, the bricks are the monomers, which are the nucleotides, which are needed to build an RNA or a DNA. Now let's find out more information about these nucleotides. Each nucleotide is made up of three parts. Now, the part is the pentyl sugar, the phosphate group, and the organic nitrogen base. So in other notes, it will say nitrogenous base. It still means the same thing. This is one brick. This is one nucleotide. There is an organic base here, which is normally bonded to the pentyl sugar and then bonded to the phosphate group. What is important to note is that the pentyl sugar is different in DNA. It will have its name in DNA, and it will also have its name in RNA. The name of the pentyl sugar in DNA is deoxyribose sugar, and in RNA is ribose sugar. We're going to speak about it. Then, in terms of the nitrogen bases, each nucleic acid, DNA or RNA, will have 4 bases. And you know this very well. When we speak about DNA, the bases that we have in DNA, it's A, C, G, and T. I've mentioned all 4. Now, I've written the first letter of the names of the bases. A for adenine. This is obviously contested every year. Is it adenine or adenine? There is no master of pronunciation. Whichever way you want to pronounce it, as long as the spelling is correct. So A for adenine, C for cytosine, G for guanine, and T for thymine. So those are the bases that we will find here for a DNA nucleotide. But for an RNA nucleotide, RNA nucleotide, we will also have 4 bases. The first 3 bases that I mentioned in the DNA, you will also find in an RNA, A, C, G. But in an RNA nucleotide, we do not find the thymine base. We actually find a base called uracil, hence the letter U. So this is very, very, very important. Then, now since we're saying DNA is a double-stranded molecule, meaning it is made up of 2 strands. When it is made up of 2 strands, there will be bases in between the strands. And these bases are going to be paired. Now, it will not be any random pairing. Whoever comes first, you can pair with this one. That's not how it works. A double-ringed base will pair with the single-ringed base. But I think that is complicated. There's a simpler way of remembering this. If you see the bases that I've written here, I've written them in alphabetical order. So A, C, G, T. Now, the first letter in our alphabetical order, when it comes to pairing, it will pair with the last letter. So meaning A pairs with T. If we have an A this side, it will pair with T at all times. Let's not worry about which one is double ringed, which one is single ringed. Then the letters or the bases in the middle, the ones that are remaining here, will pair together. C will pair with G, or we can say G will pair with C. Same applies with the bases in RNA. Now, RNA is a single-stranded molecule, meaning it's made up of 1 strand. However, the bases are able to pair when we form proteins. And in that case, where bases are pairing to form the proteins, A will be paired with U and C will be paired with G. The first letter pairs with the last letter if we are just looking at it like that in order for us to cram and remember. And then the middle letters will pair together. Now, this has explained everything that I've said. So let's look at the nucleotides now. These are still bases, guys, and this is still the introduction, and it's important going forward. Now we are looking at nucleotides. RNA and DNA are our polymers, but we need to understand that their nucleotides may differ. They will have the same parts, a phosphate group, a sugar, and a base. But I've mentioned this before. The sugar that we find in a DNA is called deoxyribose sugar, and the sugar that we find in an RNA nucleotide is called ribose sugar. The phosphate group is the same, it's just phosphate from element phosphorus. Okay, then in terms of the bases that we find in DNA, we will find A, G, C, and T. In RNA, A, G, C, and U. So that means if we were to have 1 nucleotide, this nucleotide will not have all 4 bases. We only have 1 base from the 4. This nucleotide may have base A, and then bonded to it is another nucleotide which will have base G, and another nucleotide maybe having base T, if we are speaking about DNA. So if we were to draw it here. I'm not sure if the pen is going to allow me. So this will be your phosphate group, and this will be your sugar, and then this will be your base. So the base here, for example, can be A. And then this will be bonded to another nucleotide, which will have a phosphate group and a pentyl sugar, which is the deoxyribose sugar, and another base. And this base can be T. So my point is, in one nucleotide, you're finding one sugar, one phosphate, and only one base. We move. Now, when we look at the structure of DNA, I've already mentioned the nucleotides. Now, a DNA consists of two strands. It's a double-stranded molecule. So it's two strands that are actually twisted together to form a shape which we call a double helix, a helical shape, which we're able to determine here. This is very important. When they ask you about the natural shape of DNA, you say it's double helix. If you say it's double-stranded, that is wrong, because double-stranded, you are just telling us it is made up of two strands. You are not telling us that those strands actually twist and form a helical shape. So when they say natural shape of DNA, double helix, right? Meaning it is made up of two strands that are twisted. Now, when we untwist these strands, we are able to see a ladder-like form, which will have these two strands over here. We're not going to be able to see the sugar and the phosphate group, but we will be able to see the bases in between. And we normally write it like this, and we say it's a ladder-like shape. So the nucleotides, sugar and phosphate, are still there, but we don't show them in the diagram when we just write the two strands. Now, I've mentioned this. A will pair with T, C will pair with G. Now, when we speak about the bases being paired, so if I were to have these two pairs together and I say I'm pairing them, they need to be attached to each other. There's something that needs to attach them. And there is a bond here that will attach the bases. So if I have a T here and I say the T always pairs with an A, there needs to be something to bond this pair together, and we find that bond in between the base pairs, and we call it a weak hydrogen bond. Now, in the previous years, you needed to say the word weak, but I think from the past 2 years, you don't have to say weak. If you just say hydrogen bonds, that is enough. That is very important. Now, the two strands are joined together and they will form a ladder-like structure. As you can see, these lines will obviously make the structure look ladder-like. That's it. Okay, important, double-stranded, the bases, weak hydrogen bonds, double helix as the shape. Okay, I'm going to ask, and I need people to unmute and tell me. And once I ask people a question, we don't move up until we get an answer. How many nucleotides are we able to see in this DNA structure? So I need you to look at it, unmute, and give me an answer. Anyone. I'm not going to call out people's names for now. But if people don't answer, I'm going to call out your name. Mohau?
Okay, I would say I see 5.
You're seeing 5. Okay, I'm seeing Tato typing. It's going to be difficult for me to keep up with the messages, Tato, so it's going to be easier for you to just unmute and tell me, did I hear 5?
Yes, I said 5.
Okay, 5 from Muhau. Anyone else? So I'm getting 5 so far. Anyone else?
I see 10.
Carabello, I'm hearing 10. So I've got 5 so far and 10. Anyone else with a different answer?
10.
10.
10, we're seeing 10. A few people are seeing 10. Okay, let's count them. Remember, a nucleotide is made up of a phosphate group, a sugar, and a base. So we are going to count. This is the first one, 1. It doesn't matter if you are moving from left to right, right to left, up to down, it doesn't matter. So that's 1, and that's 2, and that's 3, and that's 4. I know my pen is a bit slow. And then 5, and then we should be able to get 6 here, 7 here, 8, 9, 10. This is 9 and this is 10. So we do have 10. Okay, I need to come back. How can you see how we got 10?
Yes, I can see it, but I wanted to ask, if they are
Yes.
bonded together, don't they become the same?
I'm struggling to hear you. I'm even coming close to my laptop.
Or I was asking, if they are bonded together, then don't they become the same number?
If they are bonded together, no. So we are looking at the nucleotide itself, the one that is made up of a phosphate group, a sugar, and a base. Those three things make up one nucleotide. So even if this nucleotide is bonded with another, we can't count them as one. These are two nucleotides that involve two bases that are bonded. We can't count them as one. Okay, did you get that? Okay, thumbs up. All right, thank you so much. Okay, let's move. So I think then it's gonna be easy for us to determine this. How many nucleotides do we have on this diagram? Diagram A, and then I'm gonna have diagram B. But let's start with A. How many nucleotides here?
8.
8 nucleotides. Perfect.
8.
In diagram A, we have 8 nucleotides. Then I need someone to tell me how many deoxyribose sugar do we have in diagram B? How many deoxyribose sugar do we have in diagram B? We're not moving until we get an answer. We're not moving. We're not moving.
I'd say 16.
I'm listening.
16.
You're getting 10.
16. 1,
6.
6.
Okay.
No.
Let's see. So I'm
1,
going to take, okay, so 6. Just give
no.
me
1, 6.
1, 6. Oh, okay, thank you. Yo, I'm really struggling to hear you, Mohao. I'm literally doing this just to hear you. 1, 6. Anyone else with a different answer? Mohao is saying there are 16 deoxyribose sugars in diagram B. Anyone with a different answer? Okay, I guess everyone agrees. There are 16. So how do we count? We count the bases. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. I tried to trick you guys. I asked you about the number of pentose sugars. But remember, if you just look at the base, if you're seeing a base there, you will know that, oh, okay, this is a base from the nucleotide, meaning there is one sugar and there is one phosphate group. That is easy. So you are just counting the bases because the number of bases will be equivalent to the number of deoxyribose sugar. So that is very, very easy. Ooh, I like this part. I really like it. So to just finish off our information about DNA. Now, since we are saying DNA is a double-stranded molecule, you need to try out calculations. You need to know how to do DNA calculations. They can ask you to calculate the number or percentage of a base in multiple choice for 2 marks. That is 2 marks that we cannot afford to lose. Now, what we need to know at all times is that in a double-stranded piece of DNA, since adenine pairs with thymine, the number of adenine bases in a DNA section will be equivalent to the number of thymine bases at all times because they pair with each other. And the number of guanine bases will be equal to the number of cytosine bases. So what do I mean by this? If we're to look at this section, let's prove it. So this is a small section of DNA, and I'm going to forge my bases here. I'm just going to have 4 bases. T, I'm going to have A, I'm going to have C, and C. All right? So obviously those are my bases for one strand. I need someone to tell me the bases that will be on the other strand that will pair with TACC. Anyone? And it has to be a different person. I need more people to be active, guys. I'm going to start calling out people's names and we don't move up until you answer. Oratile, I'm with you, Oratile. Please do tell me the names or the letters of the bases that I need to complete on this strand of DNA.
And for T, it's going to, for thymine, it's going to be adenine.
Yes.
And then for adenine, it's going to be thymine.
Yes.
And then for cytosine, it's going to be guanine. And
Thank
then
you.
for cytosine, it's going to be
guanine also. All right, let's prove this. We are saying, thank you, Oratile. We are saying in a double-stranded piece of DNA, the number of adenine bases will be equal to the number of thymine bases. Let's look at our section. How many adenine bases are we able to see? 1 in this section, 2. I'm not saying per strand. I'm saying in that section of DNA, we are seeing 2. Let's look for thymine bases. How many thymine bases are we able to see? There is thymine 1, 2. I guess that is proven. The number of adenine bases that we get in a section of DNA will be equal to the number of thymine bases. Okay, we're happy with that. Let's check if that applies for C and G. Let's see. Let's see. For C, how many bases can we get? That is 1 and that is 2 bases. And then for G, we are also having 2 bases. I don't like my example. I should include more bases. It doesn't mean since we are having 2 for adenine, 2 for thymine, it will be 2 for all the bases. That it doesn't mean that. But what we want you to know is that for A and T, it will be the same amount or percentage or whatever. And for C and G, it will be the same. So what this means is if we are given the size and number of the bases in a double-stranded molecule and the amount of one of the bases, we can calculate the amount of the other. If we're given information about how much adenine is in a section, we should be able to figure out how much thymine, guanine, and cytosine are in that section. Let's do question 1. Do question 1 for me. Calculations. You are given the information. A DNA molecule contains 800 bases. This is when you work now. I've been yapping for 1 hour, 10 minutes. So a DNA molecule has 800 bases. From the 800 bases, 120 bases are thymine. Calculate the number of guanine bases. 3 minutes. 3 minutes. Then we are going to mark this together. A DNA molecule contains 800 bases. From that 800, 120 of them are T bases. Calculate the number of G bases. Tell us how many G bases are in that 800. Please do thumbs up when you are done getting the answer so that I can see how many more minutes people need. I'm giving you 3 minutes for this one. Zianda, your hand is up. Are you thumbing up or is your hand up for a question? And I think Ogutle. If your hand is up, do you have a question? Just unmute and tell me. I'm listening, Zianda.
80.
Eh?
80.
You're getting 80.
480.
480. Okay. Let's wait for other people.
280.
280. Maybe there's something wrong with my sound because I'm struggling to hear people. Okay. Anyone else with a different answer? We have 280 so far. Okay. I'm not sure if people are still trying to find out the answer, figuring it out themselves. I don't want to rush anyone, but I'll give you 1 more minute to figure question 1 out. Just 30 more seconds. All right, let's do it. Okay. I'm seeing your reactions. Let's do it. Did we all get 280? Thumbs up if you got 280. Let's see. Okay, I'm seeing a few. Okay. So let's figure this out. So they're saying a DNA molecule contains 800 bases. From that 800, you're having 120 thymine bases. So there's different ways of figuring this out, but mine is basic mathematics. So I will say, okay, they give us a total of 800. And from that 800, they're saying, they gave us T first, okay? So they're saying T has 120 bases. So this is obviously going to be easy for me because T pairs with A. So that means A will also have 120 bases. I then add the bases for A and T, and I think I'm getting 240. So I'm getting 240. So I'm then going to minus the bases 240 for A and T because that's what I know from the 800. Then I go 800 total number of the bases in the molecule minus 240 of what I know for A and T. Then I get an answer. What answer is it? Is it 560 or what? Now I have to take out my 560. Okay, thank you.
560.
Thank you so
560.
yes, 560. Now, I haven't answered my question. They want the total number of G bases. Now, what this is telling me is the 560 needs to be divided or shared by the C and G bases. And they are going to get the equal number. So I need to divide this by 2 because it is shared between C and G. If I divide that by 2, I get 280. So then it will be 280 for G bases and 280 for C bases. That's how you figure it out. Easy. Any question? Easy, peasy, peasy. Please ask me if you have a question, guys. Please. I don't like lessons without questions. Please ask me. Okay, no question. Let's do number 2. 2 minutes for number 2. Number 2 is easy. 2 minutes. So let's wait for 2 minutes to elapse. Then I will ask questions and you guys will tell me your answer. Actually need to get more water. So I'm just going to stand up and get more water. I'm back. I'm back. Okay, my camera doesn't want to turn on. Come on. Okay, there you go. Okay, I'm back. Okay, let's not waste time. Let's see. In a DNA molecule consisting of 400 bases, 10% of these bases are adenine. How many of these bases would be guanine? Julian, I'm seeing a Julian here. Julian, what did you get? Okay, there is a Lebu. I can see a few hands. Lebu, what did you get?
Okay, I got 160.
You got 160 bases.
Yes.
Okay. Thank you. Do we have other people that got a different number? So far, Lebu is saying 160. Anyone with a different number of bases? Zianda?
I also got 160.
160. Anyone else with a different number? Okay, let's look at it. Lebu, please explain how you got 160.
Okay, so basically I took the 400 bases and multiplied that by the 10%, which gave me 40. And then because it's because adenine and thymine have the same amount of bases, meaning it's 40 plus 40, which gives us 80. Then I minused the 80 from the 400, then I got 320, and then I divided that by 2, which gave me 160.
Perfect. That's how you do it, guys. So you can start by getting the number of bases, or you can figure out the percentage first. So if you want to figure out the percentage first, you can say, oh, okay, we are given the percentage for adenine and they've said it's 10%. That also means that the percentage for thymine will also be 10%. And then you add the percentages together, you get 20%. You minus that from 100%. Because the total for percentage is 100. So you'll then go 100% minus 20%, and you will get 80%. So even if you did it the way Lebu did it, it's still 100% correct. You'll get 20%. Let's say 100 minus 20%. You get, I think I'm moving too fast. My pen is not picking it up. You get 80%. From this 80%, you need to divide it by 2 because that will be the percentage that is shared by C and G. Once you divide it by 2, you will get 40%. Then from that 40%, you then need to get the number of bases from the 400. You should get 160. Clear. Let's answer number 3 and number 4, guys, so that we answer these ones and then we call it an answer. That's 5, 6, 7. Let's answer number 3 and number 4. You guys are too quiet. Are you fine over there? Are you fine? You're too quiet.
Ma'am.
Oh, hi. We're fine, ma'am. We're figuring it out. Something. Eh?
For number 3, I got 90.
Okay, I'm seeing a ooh, a you. Who's that?
Sorry, it's me.
Memo.
I can't see your name. I'm just seeing a you here. Is it a Urasil?
I got 90.
Gosnati.
My name is Tanduleti, sorry.
Tandulating. Okay. I'm thinking, okay, it's a Urasil paired with a Urasil. You know what? Anything possible.
No problem. I'm saying I
Okay.
got 90 pairs.
You're getting 90 pairs. How many
Yes.
phosphate groups?
Yes, I mean phosphorus group, sorry, ma'am.
You're getting 90 phosphate groups. Okay,
Yes.
so we have a 90 so far. Anyone else with a different answer? If you have, yes?
Can I answer? Can you
Muhawu?
hear me? Yeah, I got 180.
180.
Phosphate group.
Okay, so we have 90 and 180. Anyone else with a different answer? So we have 90 and 180 so far. Anyone else with a different answer? Okay. I forgot the name. You. Please tell us how you got 90.
Oh, so because there are 60 guanine and 30 adenine, so we already have the 60 cytosine and 30 thymine. So by adding guanine pairs and the adenine pairs, I got 90.
Okay, but then you also mentioned something correct. You said you are given 60 guanine and
Yeah.
30 adenine. Then you also told us now that means there will be 30 thymine and 60 cytosine. But in your answer, you're telling me you added 60 and 30. What about the other 60 and 30? 60 for cytosine and 30 for thymine.
You
Oh,
times
I
it,
thought as a group they are one. Sorry, ma'am.
Eh?
you need to times it by 2.
Okay, someone is saying we need to times it by 2. So why are we
Yes.
timesing it by 2?
Because of the remaining pairs that we have.
Okay, so I will take it because if you times it by 2, you're getting 180.
Yes.
Okay, it will still give you the correct answer, but the explanation will not be necessarily correct to multiply by 2. The understanding here would be if you have 30 adenine bases, that means you also have 30 thymine bases. If you have 60 Gs, that means you have 60 cytosine bases. You add all of them.
Okay.
So in this case, it will still give us the correct answer. So we are going to say 60 for G plus 60 for C plus, this is a 60, plus 30 for A plus 30 for T. That should give us 180. Why are we adding all of them together while they're asking us about a phosphate group? But we are adding the bases that we are given. Remember, in a nucleotide, you get one base that is paired with one phosphate group and one sugar. So if we are having 60 guanine bases, that means you have 60 phosphate groups. So then you're going to add the number of bases for all of what you are given to get that 180. Is it 180? Yes.
Ma'am, I
All
have
right.
a question.
Yes.
So, since they said a sample of DNA, wouldn't that only like mean the one strand of the DNA, or is just both of them?
It is both of them. A sample of DNA still means double-stranded. Is this section. So for example, if this is how long DNA is, maybe a sample is this section here. It still involves both strands.
Okay.
Yeah.
Okay, thank you, ma'am.
Thank you. Anyone else with a question? All right. Number 4. What did you guys get? Anyone? There's people that are quiet. I'll be calling out their names next week. Specifically, you've been
please
quiet.
give
There's quite a
time to-
few. Yes?
Can you please give us time to calculate? Just
I
2
must
minutes.
do what?
Give us time.
Oh, okay. Sorry. 2
you.
minutes. Sorry.
Can I answer?
Let's give the people 1 more minute. Thirty more seconds. Tato, what did you get, Tato, for your ratio?
Ma'am, I got 1:4.
1:4. 1:4. That's what you got. Thank you, Tato. Let's see if other people got a different ratio. Anyone else? Anyone else that got a different ratio, guys? There's no way that all of us got 1:4.
No, he's
Not-
correct.
Eh?
He's correct.
He's correct. It's 1:4.
Yes.
Anyone
Yes,
else with a different
simplify.
one?
Yeah, we simplified 10:40 and it's 1:4.
Hey, this group. So then we're gonna get distinctions, guys. The vibe here is giving o'clock. It's giving I'm cooking vibes. That's the language from the teenagers at work, you guys. So I'm not sure if that's the right language. So,
Ma'am,
yes,
may you please explain number 4?
number 4. All right, let's do it. If 10% of the bases in a molecule of DNA are adenine, what is the ratio of adenine to guanine in the same molecule? So let's figure it out. Let's get the percentages first for all the bases. They are saying adenine's percentage is 10. So that means that the percentage for T will also be 10. Now, when we add these two together, they will give us 20%. So we need to figure out the percentage for C and G. So C and G are going to share that 80%. How do we get 80%? We're going to say 10 plus 10 is 20. And then we minus the 20 from 100, we get 80. And then we divide that 80 by 2. When we divide that 80 by 2, we'll get 40%, meaning it will be 40% for C and then 40% for G. So then they are asking us for the ratio for adenine to guanine. So what is the percentage for A to, sorry, not percentage, the ratio for A to G? We can start by writing the percentage. We said A is 10% and we said G is 40%. We don't leave it at that. We simplify it. Someone did say it here that we need to simplify it. So when we simplify it, I'm going to do it the 1995 way. How many times you think, okay, which number is the smallest one between the two is 10. How many times does 10 go into 10? It goes once. How many times does 10 go into 40? It goes in 4 times. That is your ratio. And remember, when you write a ratio, there has to be a colon between the numbers. No comma, no fraction. So you can't write 1,4 or 1 over 4. That will be incorrect. You can't even write that. Okay? I'm hoping that is clear. Is it clear? Yes,
Yes,
yes.
ma'am, it is. Thank you.
Okay.
Yes, ma'am.
Thank you so much, guys. All right. That's it for today. But please don't leave the meeting yet. That's it for today. I will send 4 more calculations on the group just now. It's up to you whether you want to answer them today or tomorrow, but I will send the memo on the group around, I think, over the weekend. So please do them. It's just 4, and that's it in terms of our homework activity. In the next lesson, we are going to dive into the real business, DNA replication, and I then send you activities from previous question papers. And then we do protein synthesis. I do the same. Questions from previous question papers. And then we are going to do DNA profiling. We close the topic. Then we move to meiosis. So we are going to try and do questions from previous question papers up until June exams, and we get ready for the exams, and then November exams. So we are going to take it week by week, and I really need us to be one big happy family. That's what that WhatsApp group is, our support group, guys, for questions. Even if, let's say, now you are busy with protein synthesis. I know last week there was someone that asked me a question about transcription. Feel free to send a question. Let us be there for each other. Let us help each other. This group is for us to really be there for each other and, you know, to get the good marks that we really want to work hard for. So that's it from me. I will send the homework activity now. Weekends, there's normally a weekend checkup to check if we've done the work. And I know you can be probably busy at school and with sports and all other things. So I totally understand that. But please do not cheat and just wait for the memo. That's not learning. Okay, the house rule here is before you leave, you need to unmute and then you say bye. Then you can bounce.
Before we say
Yes,
bye,
yes, yes.
do you offer the classes in English only?
Yes, they are in English only.
Okay.
Okay, you needed them in a different language.
Yeah,
Which is?
Afrikaans.
Ek nie praat nie. Nee.
Can I also ask a question?
Sorry, basically, Agni Pratni. Sorry.
It's fine. It's fine.
Okay. Yes, someone wanted to ask a question?
Yes. Is 5:30 the only time we're going to do these classes on Wednesday?
Okay, that was the time that I suggested, and there was no one that said, I think there were only 1 or 2 people that said they might struggle with 5:30.
Yes.
So I
Yes.
did see the message on the group. There was no way I could change it today because I work on a schedule myself. So I can't change things last minute. So if people are voting, I'm going to put a poll now or tomorrow. If people are voting for 6 o'clock, then I'm happy for us to start at 6. But then it might become a problem if 50% say 6 o'clock and 50% say 5:30, because all my messages are saying 5:30. But let's put up a poll and then see and take it from there. Is that fine?
Yes, it's okay.
Okay. Thank you, guys. Anyone else with a question? We all good. All right. We unmute and say totsins and then bye.
Thank you.
Thank you so
Bye.
much.
Thank you.
Thank you. Bye
Bye,
bye.
guys.
Bye.
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 Yeah,
Bye.
Sure. Bye.
Thank you for your time.
Thank you for your time. I really appreciate you
Bye.
joining. Bye, guys.
Sure.
Well, you click on leave. There
Oh,
should be a button that says leave the meeting.
okay, sure.
Unless you don't want to leave the meeting.
Oh, we are still continuing. It's
No.
fine.
Bye, guys. Bye. I'm going to leave the 3 people here. Litaho. Okay.